Hi, madam. My name is Muhammad Fishikin bin Sukaran. I'm from group 80205B. So today my topic is propagation of pineapple, sexual and asexual. So this is my overview. The pineapple plant is generally reproduced using vegetative material and asexual reproduction with no new genes combination. However, asexual reproduction is possible since the seeds are formed in the fruits when cultivar cross-pollinates spontaneously or under controlled setting. So without further ado, let us go to the first one, which is the asexual propagation. Pineapple plants are usually propagated using vegetative planting material. Uh, there is a wide variety of type of vegetative material and both cultivar and environment effect the availability and quality of material used to plant commercial field. And the more common type of material produced naturally by the plant, also referred as to conventional planting material, including the first one is seeds, the leafy branches attached below the fruit on a pedicule, gathered towards the fruit base and sometimes formed from the fruit bacilli. The second one, which is cola. These structures are generally preferred and can produce fruits 14 to 16 months after planting. And the third one is ground sucker or ratoon. Fruit will be produced 12 to 14 months after planting by shoot formed from the stem slightly above the ground, and side shoot or sucker. The fruit will be generated by shoots from the above ground selection of the stem 18 to 20 months after planting. And the last one, which is will be crown. The short stem and leaf that grow from the fruit apex are not often used. The fruit production might take up to 24 months after planting. So the non-conventional planting material may be obtained under field or nursery condition by human interference applying specific techniques. So there are four techniques in asexual propagation. So the first one is the plantlets obtained by the section part of the plant, which enable the development of lateral buds present along the stem of the mother plant or the small stem of the conventional type of planting material that are mentioned above. So this is the second one. Plantlet are forced to develop by destroying the apical growing point of vegetative part or gogging and simultaneously growth of lateral buds. So this is the third one. Plants or propagules form instead of flower and fruits by plant treatment with chlorophenol at flower initiation. So this is the last one of asexual propagation. Finally, there is a method of micropropagation, a multiplication of pineapple in vitro, a technique of great utility especially for the rapid multiplication of a new cultivar produced in a breed programs. So this is the sexual propagation. In traditional genetic improvement system, breeders carry out manual cross-pollination between selected pineapple plant of a different cultivar during simultaneous flowering period synchronized by forced floral induction. Opening of flower occur in a over period of about 3 weeks, and at every open flower are pollinated. At harvest, seeds are separated from the fruit pulp, cleaned of fruit residue, dried and stored for cultivation. Pineapple seeds are small, averaging about 5 mm length and by 2 mm in width. They have a brown, very resistant leathery integument and hard and fine of endosperm. So the asexual propagation. With the onset of reproductive stage, new leaves stop to develop and previously starred leaf fail to expand to full size. The brake is the first structure to develop and the fruitlet exists, followed by the primordia of the sepal, petal, and stamen. The carpels are the last to develop. The pedicule begin to expand. As the first petal appear, the flower primordia grow to ascending order as a new brake appears above. And the number of floweret generally and inflorescence varies greatly according to the variety, size of the plant at the induction, and the plant population density. Within 3 months of seeding, seedling have a 6 to 8 leaves and move to the pots of agriculture substrates and nutrient in the greenhouse or screen house. After an addition 9 to 15 months of growth, during which the seedling reached a minimum size of 25 centimeter, which is considered adequate for plant in a field. The plant are finally accumulated in an open nursery. During this cycle, plants are initially screened and selected based on a variable such as the presence of leaf, spines, leaf color, growth vigor, fruit shape, size, and internal qualities. So this is the conclusion. So to conclude this, pineapple is commercially grown through vegetative method, which is restrict its multiplication rates, a feature that contributes to a slow increase in area planted and output of this fruit. Technique for propagation using traditional planting material were primarily adapted 30 years or more ago. With a little new knowledge provided in recent years, this century research has mostly concentrated on improving quick propagation uh, through the plant stem selection under nursery circumstances, especially micropropagation. The former is important because it must be continuously adjusted for the new cultivar, whereas the latter must also be studied uh, further in order to reduce the cost and the improve of the economic performance of the planets. A critical condition for the future worldwide is used for this propagation method for establishing commercial pineapple field. So that's all for me. Thank you.